Hello, my name is Preethi. I recently graduated from UC Berkeley with a bachelor's in molecular and cell biology and a minor in German. I've been a research intern here at the Regenerative Sciences Institute for about 6 months now. As part of the AMRITA Initiative, my goal here is to rejuvenate old differentiated human cells using techniques from synthetic biology and also bioinformatics. I've been working on several projects since I started working here. For the first project, I successfully transfected 293 T cells with novel gene therapy vectors to study their utility. This project gave me a solid understanding of all the techniques involved in cloning from start to finish, as well as mammalian cell culture techniques. I also learned how to use Gentle and ImageJ, a DNA analysis software and an image analysis software, respectively. I've also been working on a couple of bioinformatics projects involving enzymes involved in maintaining and regulation of our epigenome. Methylation and demethylation play a big role in the epigenome. The first project involves studying the evolution of the TET1 protein in various organisms. TET1 is involved in the process of demethylation. So once I found an interesting domain in the Homo sapien TET1 genes, I then proceeded to search for other proteins that interact with the domain. This seemed to lead to a dead end. However, I'm planning on reading on more literature on the topic to further my research. Another bioinformatics project I'm working on involves studying the evolutionary history of multiple proteins involved in methylation to discover unique characteristics that may potentially affect their function. I discovered that the SETMAR gene in Homo sapiens has a transposase domain in addition to the methylase domain. Transposase is an enzyme that catalyzes the movement of a segment of DNA from one place to another place within the genome. They're also known as jumping genes. I've been trying to determine the places the SETMAR binds to in the human genome and also try to profile the neighboring genes in terms of their function. For the bioinformatics project, I used various NCBI tools such as the BLAST, taxonomy browser, and also the conserved domains database. I also benefited from using websites such as UniProt, PFAM, and Interpro. I also used various protein interaction databases such as BioGrid, Mint, StringDB, IntAct, and Scikit. I did literature searches using PubMed. I used various softwares such as JALView, BlastViewer, and SysGenome. I am self-taught in Python and I'm able to code at a beginner's level. In addition to the bioinformatics projects, I'm also simultaneously working on 2 projects involving wet lab techniques. The first project involves trying to rescue old differentiated human cells from senescence. It's a kind of cell aging. However, I don't wish to revert them back all the way to the stem cell stage. Senescence occurs during a cell's life cycle when the cell stops dividing and the activity starts to slowly decline. Senescence can be caused by multiple factors, including telomere shortening, stress, or even after a set of cell division. I hope to revert them using viral particles that carry the Yamanaka factors. Yamanaka factors are a group of 4 proteins that induce pluripotency in mouse and human cells when highly expressed. For this project, I learned to produce and concentrate viral particles and also determine the titer of the virus using a titration assay and also an ELISA assay. The second project that I'm working on involves carrying out in vitro coupled transcription translation reactions. For this project, I've been following the method from a paper. I extracted the cell lysate from 293 T cells. For this project, I learned how to work in an RNase-free environment, use the size exclusion chromatography column, and also use a spectrophotometer to read the absorbance of the cell lysate. So overall, this experience has been very rewarding for me. In addition to strengthening my lab techniques, this experience has really allowed me to grow. I've learned to be more proactive, so whenever I start an experiment, I try to understand all the concepts, all the techniques, and I also make sure that we have all the reagents before starting the experiment. This experience has also nurtured my troubleshooting skills. Whenever my experiment goes wrong, I have to think of things that could have gone wrong. Troubleshooting is a learning process. There are so many things that could go wrong in science. But I've learned that while it's okay to make mistakes, it's important that I learn from them. In the future, I hope to go to graduate school and study in the field of bioinformatics.